Welcome to episode 7 of BioTech Talk, your one-stop show for all the tidbits of life sciences and biomedical industry, powered by Lita Life Sciences. So today we have a very interesting person with us, Mr. Mehdi Mobarek. He told me not to say Mr., but I think it comes with the flow of it.
That's
But
all right.
so I will keep saying Mehdi Mobarek. Welcome, sir. I'm very grateful that you have taken your very precious time out for our wonderful show to talk about very specific things and very interesting things in biotech talk.
Thank you very much.
Are you excited to be here?
Thank you very much. I appreciate the invitation. It's a first for me. Like I was saying just a few minutes ago, I'm an action man, not a showman. So that's a first podcast for me. But thank you again for inviting me. I appreciate really.
Absolutely. But he says that he maybe with a mic he's doing it for the first time. But I do not believe that he's not a showman because the moment he gets in front of people and talks about his topic, he's a killer. So Mehdi is actually the CEO of Agenta Life Sciences International. It's a UK-based company, and it's kind of headquarters right now in Dubai as well.
That is correct.
Yeah, and they basically support research solutions, and we are really proud to have them as our exclusive partners as well.
Thank you.
Yeah.
Yes, indeed. Agenda support the research in general by deploying experts from Western countries to the Middle East region where I'm located right now. So it supports all kind of laboratory research from in vitro to in vivo solution, which is the subject today.
Yeah, exactly. So we briefed him quite well in advance because Mehdi is also someone who likes to talk. He is someone who can speak for 2 hours or 15 minutes, depending on what the requirement is. So today we have actually 10 minutes.
Okay.
10 minutes to speak about our topic, which is...
It would be good enough.
Really?
Absolutely.
Are you sure?
I'm 100% sure.
100%. Amazing. Okay, so today's topic is From Models to Medicine, Transforming In Vivo Research.
Great topic.
Let's start.
Let's start.
Okay, great. So we'll start with the basics. Like all the episodes, we start with the first line of the thing. What is in vivo research, and what does it entail, and what is it all about? Basically, like we got a brief outlook of it from the previous episode, but we never really got into understanding what exactly it is.
Okay, that's a very wide question. To respond to all of them in 10 minutes, I'll try to make it short. In vivo research, you already have two words.
Okay.
Research and in vivo. I'll start with the word research because research means a lot in many different fields. It could be mechanics, bioengineering, biomedical, and then we're going to focus more on biomedical, which is our subject today. In biomedical, we distinguish in vitro research and in vivo research.
Okay.
Commonly, in the Middle East region, people are more into in vitro research.
Okay.
In vitro research means working with individual cells, protein, DNA, tissues.
Okay.
All in a lab, in a dish, in a tube. That's why it's called in vitro.
Okay.
And this is commonly known, and it's a very important step in research. Now, to focus on my specialty, which is in vivo research, in vivo, as the word says, it means live models. Live models, more commonly, it's a use of animal models for the research.
Okay.
That's what defines in vivo.
Okay.
You need a combination of in vitro and in vivo to succeed in any kind of research, from the drug discovery all the way to treatment. And when I say treatment is a wide term again, as it touches not only human, but it also touches animals. So we use animal models for animal diseases, to cure animals for the animal welfare, animal well-being, as well as for humans.
Okay.
And we use it in many other fields for chemical testing and others, but we're not going to go to this subject right now. So in summary, in vivo means live animal models to be used in research.
You mentioned animal models. So, is there different types of models that caters to different researchers?
That is a very, very good question. The reason why we use animal models is because we have different kind, as you mentioned.
Mm-hmm.
It starts from zebrafish,
Okay.
Medaca fish, some little fish,
Mm-hmm.
all the way to large models.
Okay.
When you say large models, it can go all the way to sheep and goats and everything. There are ways of defining which animal you want to use.
Okay. Mm-hmm.
And people say, why do you use animals?
Mm.
There is multiple reasons why you need to use live models. I'll keep the animal side for a minute.
Mm.
You need to use um, live models because you need to, after you've proved your concept in the lab,
Mm-hmm.
in the
Mm-hmm.
tissue, in the cell, if you're targeting a protein, you're targeting a function
Mm-hmm.
of a receptor or anything, then you need to make sure that your drug, your treatment, your analysis is valid
Mm-hmm.
on a live model.
Okay.
Why? Because you have, in a live model, you can follow the administration,
Mm-hmm.
the diffusion,
Mm-hmm.
the metabolism, and the elimination of a drug, an injection, anything that you will actually work toward the treatment.
Mm.
And it's not possible to do that on an individual tissue or an individual cell.
Oh, yeah.
You prove concept in vitro,
Mm-hmm.
you confirm in vivo. And that's what's called the preclinical research.
Okay.
And the preclinical research means the before the human clinical trials, which is common.
Mm-hmm.
So we do not take a risk of giving medicines to humans or even to animals without having tested it on laboratory animal models.
Okay.
And I insist on the word laboratory because those animals are bred, are genetically designed for research. So you do not take animals from the field or from anywhere in the house. You don't take a mice from a house and say, okay, I'm going to test on it. It doesn't work this way. There is a lot of more regulation, ethic toward how to use animals in research.
We are talking so much about live models, animals, and you just said that we don't pick up the animals from the field and they're bred separately and specifically for this purpose. But you did mention the ethics part of it. So, isn't there a common people concern over the ethics of using live animal models, be it even if it's just laboratory specific one? Can you care to weigh in on that?
Sure. The general public is rightly concerned because we all have pets at home.
Yes.
We all love our animals.
Yes.
No one wants to see an animal suffer.
Absolutely.
And it's a mistake to think that research makes the animal suffer. The use of laboratory animal science is extremely regulated worldwide. And there is government regulation, but there's also ethical committee on each laboratory, which is mandatory. So in order to be able to practice research on animals, there's a lot of stages to know. First, it starts with the right infrastructure.
Okay.
So you cannot put animals in your kitchen and thought, okay, I'm going to test this or this food. Because testing is not just on drugs, it's also on food allergies and others.
Yes, yes.
So you have an environment to control. And then from the environment, you have the people knowledge to control. So there is different employees starting from the husbandry of the animals all the way to the people allowed to practice invasive research on animals. That means doing a surgery or an injection. Those people have to have certification and training. And the training could last 1 year, but for most, it lasts several years.
Wow.
So this is how much the biomedical research cares about the animal welfare and animal well-being. In the use of animal, you said the animal models, for the general public, it's still a very wild word. How do I know if I need to use a mice, a rat, a rabbit, or any other species?
Yeah.
Well, it's based on the disease you're targeting. It's based on the genetic model. Because when we say mice, most people don't know that there are thousands of types of mice. We genetically work on a model to develop a specific disease. So if you're targeting obesity, diabetes, cancer, Alzheimer, Parkinson, there are different kind of models.
Okay.
So you don't take just a animal and said, okay, let's see how it works on it. It's far beyond that.
Yeah.
And this is what the laboratory animal science is about, is the knowledge of using the right animal for the right diseases. And it goes again from picking up on the fish, and you would say, why fish?
Okay.
because it's easier to see through the fish as it's transparent.
Ah,
So you can follow some of the functions.
Mm-hmm.
The fish reproduce itself more.
Mm-hmm.
Why mice? Why we share, as humans, we share 95% of our genetics with the
Wow.
mice.
Wow. With the mind.
People don't expect that.
That-
Same as when you talk about the HLA. I'm an immunologist, so HLA,
okay.
HLA on the pig is
Hmm.
common. Is when you talk about organ transplant.
Mm.
Well, the pig organs are the closest,
Oh, wow.
and so on for the different kind of animals. So because we only have 10 minutes, like I said, I can speak hours on
Yes,
that topic.
very much.
But just to reassure that there is a lot of ethics, a lot of care. People who work with those animals love the animals as much as you and I love
Yeah.
our pets.
Our pets.
And there are protocols,
Hmm.
SOPs on how to handle the animal models. Basically, we put the animals at the best comfort.
Mm.
First, because we like the animal, but also it's important for science because science is about repeatability.
Yes.
It's about the environment. Any difference in environment might affect the result of your research. So you want to control the environment. You want the animal welfare and well-being. So you don't want stressed animals. You don't want suffering animals. So we take care of all that to make sure that the research deliver what's supposed to do, which is the right treatment, whether it's for the veterinary science or the
for
medicine for humans.
Human science. So that's very well put, and I hope that people do understand the conundrumic issue here, and then understands that it's not about sacrificing any of the lives.
Correct.
It's
I will add one thing. Sorry,
Yeah.
I forgot to mention. In the laboratory animal field, we have what we call the 4 R's. It used to be the 3 R's. Now it's 4 R's. Replace,
Mhm.
refine, reduce,
Yes.
and respect.
Oh, wow.
This is the rules for all the researchers working in animals. And the word is self-defined.
Yes.
We reduce the use of animals whenever we can by using alternative models.
Alternative models.
We respect the way we use the animals. We replace whenever it's possible. We reduce on the protocol. So we do everything possible to use less and less animals because the goal is not to use animals. The goal is to find treatment, the right treatment, and to treat well the animals that help us define the treatment for the world.
Understood. Yeah, that's very well put, actually.
Thank you.
So, how does the researchers or scientists, how does this in vivo come into play in the collaboration of researchers or scientists?
As I was mentioning, we talk about in vitro first. It all starts with a cell, a piece of DNA, a piece of RNA, which you extract using different methods, different technologies, which Leader Life Sciences is very well aware of, as involved in the different steps of the research.
Yes.
But then when you have a concept, you move into the animal model.
Yes.
In between, you have different core of research. Starting from a chemist, when you talk about a molecule or you want a compound that has an interest, extraction from a plant, from a mineral, from anything. So it's from the chemist all the way to microbiologist, to biologist, to immunologist, to behavioral study. They all need to interact because they all use the in vivo to prove what has been discovered in vitro.
Okay.
So the researcher collaborate. If I take an example how an animal research facility work, because this is where we keep the animals for different researchers. So first you have a platform breeding, maintaining animals for different kind of research.
Okay.
It could be from metabolism, pharmacology, toxicology, immunology, so all kind of researchers. But how they collaborate, they start with the technician who actually do the husbandry, who knows about the animal behavior, because every animal behaves in a different way. You have a veterinarian, a vet doctor, that has to make sure that the animals are in the right condition. So this veterinarian discuss with the researcher to actually target specifically their research program. So the collaboration is always there.
Okay.
It starts with a doctor. When you go to a hospital and you expect a doctor to be cured, this doctor usually does research on the side.
Okay.
To make sure that he has the latest treatment for cancer, for example. This doctor has to collaborate with the vet, which has to collaborate with the animal technologist, which has to collaborate with many other specialities. So in research, it's all about collaboration. Furthermore, if someone finds an interesting treatment in Dubai or in Stockholm
Mm.
or in Paris or in London.
Mm.
The repeatability of the study is to be able to pick up from whatever the researcher started, because we're talking about standards.
Yes.
This is a very important thing in science.
Mm.
Standard. We use the same standards, whether it's ethical, scientific, health standards,
Mm-hmm.
and then people develop. So you can pick up a research from US to China to Japan to Middle East and continue on. And that's the beauty about science.
Mm-hmm. It's a very intricate system, starting from the doctor that you visit to the end animal researcher that goes into play. It's quite fascinating how this model works. So, what is the current, like, just to drive it down the show, the conclusory statement? What is the current situation in GCC? Like, how is it? Is it easily adopted? You did mention in the beginning that GCC is more driven towards in vitro, and do you see a future of in vivo model adoptions in this region?
Absolutely. It's a must.
Yes.
I've been in the Middle East. I mean, I've been in this field for more than 2 decades.
Yes.
And more specifically in the Middle East region for the past 5 years.
Mm-hmm.
And I've seen already a development. Like I said before, the GCC region is very focused on in vitro because of the lack of availability of animal.
Okay.
It's not, like I was saying, you don't pick up an animal from the field
Yes.
and grow it. The animal models are mainly Western bred,
Okay.
under licenses. So it's all very, very controlled. It's one handful of companies that can provide the right animal models to the right genetic, to the right everything.
Yes.
In the Middle East, obviously, they started with the in vitro, but didn't get really all the way to the in vivo. In the past, I would say past year, I'm seeing an activity back on people wanting to start the in vivo model, because they understood that one platform can serve 50 research units.
Yes.
And they all need at some point, if they want to have the autonomy of developing drugs, and more specifically to the population, because we're all genetically different from one part to the other. So a drug developed in the US might not work the same way in India or in Middle East or China. So it's depending on the population. And for that, you need to do local research.
Yes.
This is what the GCC region is starting to do slowly, I would say. I would hope that things move a little faster, because the ground is there, the infrastructure are there, the knowledge of some of people are there.
The intent
is there.
Yes.
Now is to put everything on the same platform and offering. And this is the main reason why I decided to deploy Agenda from the UK base to the Middle East. So basically, Agenda support the research in one of the most famous universities worldwide, without giving names. And we're looking at providing the same in the Middle East, because there is no reason why it cannot be transferred. It's transfer of knowledge, sharing of information, sharing of knowledge.
Yes.
As a summary, I would say there is no research without in vivo.
Absolutely.
As simple as that. So you need the in vitro, you need all the extraction, DNA. There is a lot of fields to talk about, but all of them need in vivo, because at some point, how do you validate a drug?
Yes.
A vaccine. We've been through a pandemic not long ago. How do you think this was developed?
Yes.
General public just wait for an injection. But this injection is years of research.
Yes.
From in vitro to in vivo. And we are in the GCC on the stage where we do need now to make it happen in vivo, because there is no obstacle to it. I hope I've responded to the topic as expected.
Yes. I think it's also nice to see that, like how you say, without in vivo, there's no research. So it also speak to the need of the GCC to be self-sustainable. So I think that kind of addresses that as well. So it's really great to have you here and speak really very informatively and concisely about a very vast topic, I'm assuming. So I'm
trying to be concise. I'm so passionate about the this field of laboratory animal science that I could go on for hours, but it's important for the general public as well, for the educated people to know it's simple when working together.
Yes.
And it's a mix of different expertise. Everyone brings its expertise from in vitro to in vivo. Hopefully I'll help as much as I can in the in vivo side of things.
Absolutely. With with that note, we'll come to the end of this show. Thank you for being here. Thank you, the audience. I hope you had a good time as well listening to this amazing man speak about laboratory animal sciences. So, this is episode 7 of Biotech Talk, powered by Leader Life Sciences. Malavika Kanara signing off.